My name is Prince Saforo Amponsah. I am a scientist in the lab of Bruce Morgan at the Department of Cellular Biochemistry in the University of Kaiserslautern in Germany. Our group is interested in understanding the fundamentals of redox biology in the cellular context. Our research focus is basically twofold. First, to understand how redox species are generated and regulated. And secondly, we are interested in how changes in specific redox species affect cellular physiology. To address our questions, we develop and utilize novel genetically encoded redox sensors. These sensors typically consist of an endogenous protein which acts as a transmitter of the redox signal, coupled to a redox-sensitive GFP. So changes in the cellular environment is detected by this sensor and transmitted to this GFP, which results in changes in fluorescence. We make use of the CLARIOstar as the routine instrument for fluorescence measurements in our lab. So the CLARIOstar is very simple and easy to use. We are able to run our assays in a 96 well or 384 well formats. This means that we are able to do our experiments in medium to high throughput, measuring several strains and using multiple sensors simultaneously. The CLARIOstar is very important to the redox community because it's a very sensitive instrument and can detect even baseline sensor signals. Compared to other instruments, the CLARIOstar has the advantage that when strains have different expression levels of the sensor, it's able to adjust the focus and gain of the sensor signal across the whole plate to accommodate these differences in expression of the sensor by the strains. In the future, we are looking to understand the crosstalk between redox species in different cellular compartments. And what we are doing at the moment, together with other labs, is to develop sensors with different fluorophores and target them to different cellular compartments in order to be able to monitor redox species simultaneously. And this would give us an idea of how there is crosstalk between these species and how they influence cellular behavior. For us, the CLARIOstar is very important because it's very simple to use, it's very robust. The CLARIOstar is sensitive enough that even without chemical perturbation, we are able to detect baseline sensor signals. For the redox community, this is very, very important. Now, the monochromatic base system means that we don't have to spend extra money to buy filters, but we can measure at multiple wavelengths using multiple fluorophores, screening multiple strains simultaneously. This is very cost effective, and this is very important for the redox community.